Take two. We're going to try this a little different. Still experimenting. You guys let me know if this even works well for you in this format. This is not using the MSU recording room, so tell me how this goes. I got to speak fast, got 15-minute increments, and this requires me to stay short. So we left off with the heterochromatin. Do go through these bullets just to see how it differs from euchromatin. Again, we're interested in these regions of the sister chromatids that are less bound. So if we go up a slide, oh, come on, are you serious? There we go. You might remember this would be akin to the heterochromatin, unusable, tightly coiled. This would be more like the euchromatin right here. It's exposed, it's open, it's available for binding by other proteins to be read, synthesized, transcribed, or used. So we're interested in the euchromatin, not the highly dense heterochromatin. That's usually the form it is when it's shut off, not being used. So those are gene poor areas, places like the centromeres, near the telomeres, highly repetitive sequences like simple sequence repeats, the SSR, transposons, things like this, which could also be mutated but doesn't have any real effect on the genome or the phenotype of the plant. So let's go to the gene-rich regions and look at the genetic structure. So these long strands of DNA, you have the nitrogen-based pairs, you saw this slide, and it just continues on and on, right? And in there somewhere, there is a specific sequence or code that kind of gives direction and instruction. There are programs and algorithms that bioinformaticists will use and will highlight these regions, and we can request, say, from labs that, let's say we're interested in this blue region right here. And we can give them this sequence as a primer. They'll make it. And so any DNA that has this sequence, we have an enzyme, a primerase, that can bind to it and begin to read with the DNA polymerase II. This is what PCR does. Again, that's all stuff for biotechnology and deeper study, but just to show you how we can, what looks like a bunch of nothing and just letters, long sequence, actually has coded information in it. So what are those letters? So no longer looking at letters, just genetic structure. Here's a typical gene right here. This is the one we went through in class. We can say this is for lactose utilization gene. So if we wanted to break down lactose, don't pay attention to the actual example, just look at the elements, okay? So here's the DNA strand. Along that sequence, you could have a regulatory gene that is a gene that codes for a protein that gives no trait. It's a protein that regulates the use, the turning on or turning off of other genes which do serve a function. So there are sequences in the genome that don't code for a trait, but they do code for a regulatory protein. This protein would bind to another genetic element called an operator. So this sequence, whatever this yellow sequence happens to be, this is what the protein specifically will recognize in only that region. And when it's bound to that region and present, then this other protein, RNA polymerase, which would look to read the genes, cannot bind to it because it's blocking a part of the promoter. So we have sequences which serve regulatory functions, such as an operator. We have promoters, which precedes all genes here. Typically, in eukaryotes, it gets pretty funky. This is a simple example, though. That promoter region is not always located right here, but the point is you have these segments, which are non-coding segments, but they do serve a function in helping or repressing the expression of these three genes, which if these three genes were actually read and expressed, would result in a protein that's an enzyme in this case that would break down lactose sugar. So we're only really interested in the labels to make that point. Here's another way to represent it. Again, the DNA strand, and along that DNA strand you have a regulatory gene, possibly a binding region for the protein that's a result of this gene would bind to it. Here's a promoter. This whole structure from promoter, operator, And the structural genes themselves, what we actually think of when we think of genes, are these right here. That whole region is a unit called an operon. That you want to know. All right, so when I continue to make reference to expression, now we're getting into the next topic. So this is going from genomes to genetic structure and the central dogma of biology, or gene expression. You might have also heard it referred to as protein synthesis, because ultimately the result of this is to synthesize a protein that allows that cell to do something it wasn't doing before. There's no reason to make a protein unless that plant needed to do something, carry something, break down something. It could be a transport protein for moving vesicles from cell to cell through the plasmodesmata. For viral transport, sometimes viruses will hijack these proteins. For pulling in sodium or nutrients from the soil across the root cell membrane, you need proteins to do that function. So those are what we're referring to with structural genes. They actually perform a function in the cell. And expressing this gene means making a protein, which is why protein synthesis, gene expression, and the central dogma are all synonymous as far as we're concerned. All right? So the first thing that has to happen when you have a structural gene is, assuming all operators and promoters are either repressed as needed or activated as needed, then that RNA polymerase, just like DNA polymerase, was able to synthesize a new DNA strand, RNA polymerase is able to synthesize an RNA strand. So here's that messenger RNA. So one form of RNA is mRNA. And so once this unzips and unwinds, RNA polymerase makes a single strand. So DNA is a double-stranded helix. Messenger RNA is a single strand that is a complement to whatever these genes were. And this is called transcription. We've transcribed the message. We've simply rewritten it. Messenger RNA then is what typically leaves the nucleus and is translated by ribosomes. And ribosomes will pull in amino acids according to the mRNA code and make a protein. Don't worry about, I'm always giving you the current and looking ahead, so we'll slow down for this. We'll go into detail in the next lecture or next time I see you. I want you to notice is A, B, C, and D. Those are 4 different subcomponents of the single gene. So oftentimes you need a polypeptide, which is a protein made of many different amino acid strands. So A might be one polypeptide, B, C, and D, which in and of themselves may perform a function. But in this case, it requires the presence of all 4 polypeptides, otherwise known as proteins, to be together to perform the function. So when I have these arrows coming from this, it's because A codes for protein A, gene B codes for protein B, C, C, D, D, which is covered by my picture. Oh, there we go. Check that out. Learning something new all the time, man. Next. So same picture, well, same concept, new picture. So here's a DNA double helix. The top, again, just tell us that there's many things in the code. It's not all structural genes. So here's an enhancer region. An enhancer does exactly that. Some genes in a plant are always expressed, constitutively expressed, just at some pace. You're always going to be doing photosynthesis, right? But the plant can detect when daylight is longer or shorter, and that light becomes an impetus. And enhancers can cause, instead of going, say, zzz, zzz, and my zzz is simply imitating, that helicase or RNA polymerase going right down the DNA line, making the gene into a protein. Well, with an enhancer present, like seen on the screen there, it just makes it go so much faster. You make so many more copies of that gene for more protein, especially if you're in a developmental stage where you're growing. So young in development of a seedling, just as you would as a baby or an embryo, these enhancers really turn the gene on. So here's the actual gene. Here's the upstream elements, genetic elements. This is downstream elements, and you have enhancers. Enhancers can be on either side of the gene, and you'll see why here in just a few moments. So if we look at the gene itself, what we now consider a gene, we're expanding our definition. You'll notice it has a primary transcript. So this would be the messenger RNA, mRNA, but before it's the real mRNA, we call it a pre-mRNA or immature mRNA. That's because it needs to get the cap. But notice these new elements that I'm introducing now, exon and intron. Now, obviously, there's many introns and exons in a gene. This is just for illustration. Introns need to be snipped out. They're intervening segments, again, that mean nothing. They're spacer regions. Why we can talk about would be very interesting to genomics, but for now, for the function of a gene, we just want to know that it needs to be snipped out of there. So we're going to snip that intron, and we're going to splice together the two exons. All right, and you can see that right below. So splicing. So we've rewritten the genetic code, but as it turns out, not everything in those structural genes actually codes for the protein. There are some intron regions. The exons are spliced together, and now we have all that is needed. So this is really going to throw you. This is pretty cool stuff, and I'll talk about this again. Let's look back at this, A, B, C, and D. As I mentioned, in this case, maybe all four proteins were needed to have a functional protein, but there are other functions in the cell where just A or just B, or it could be ACD or ABD or BCD or ACDC. Yeah, see, it just depends on what message you need. Okay, corny teacher joke. I tried. Not even a joke. So going back to the splicing, these can be spliced in any order depending on the genes. So already with very limited information, we can do many different things depending on how many of those polypeptide strains we want. So here's a gene, has exons and introns. Introns need to be removed. Exons need to be spliced together. Now you have a mature messenger RNA with a cap and a tail that we may talk about more later. And this is what's going to help us to get out of the nucleus in a plant cell into